So hello all of you. So in today's video, we will be discussing uh, the solution or the questions related to the GATE Excel or Life Sciences questions. The topic from where we will be discussing today's questions is biochemistry, structure and function of biomolecules. So to begin with, this is our question. So this has come to one of the GATE question paper in the previous question paper. So here it has been asked that which one of the following amino acid is responsible for the intrinsic fluorescence of proteins. The options provided are A. Proline, B. Methionine, C. Histidine and D. Tryptophan. Okay. So to answer this question, first we need to understand the amino acid structure and their side chains. So to begin with, so the basic structure of amino acid, that is the alpha carbon and all the four valencies. So here amino group is added, another portion a carboxyl group is added, hydrogen is added, and here R group is added. Okay. So R group could be any other side chain and which brings the diversity in the different amino acids. So this carbon of alpha carbon of amino acid, as all the vacancies are possessed by four different groups, this carbon is chiral carbon. Okay, except in case of glycine where this R group is hydrogen itself. So in that case we'll see the glycine structure. Okay, so there are two hydrogens attached to the alpha carbon, so only the glycine structure is only in the glycine structure this alpha carbon is not chiral, it is a non-chiral compound. Except glycine, all other amino acids comes under the or have the alpha carbon as a chiral carbon center, and because of their nature or of because of this chirality, all the remaining amino acid except glycine are optically active. Yes. Okay, so this is the basic structure of any amino acid. So there are different R groups, and based on that there are different amino acid classification. So so this is the whole 20 found amino acids. Okay, so here this group of amino acid where non-polar aliphatic R groups come. Here you can see glycine, alanine, valine, leucine, methionine and isoleucine. So there are different hydrocarbon chains attached in place of R group in the uh, amino acid backbone. Now then comes another group which is polar but uncharged R groups. So under this category we have serine, threonine, cysteine, proline, asparagine and glutamine. Other than this, we have our another group, another three groups, that includes aromatic R groups, positively charged R groups, negatively charged R groups. And among these amino acids, there are two which contains sulfur in their structure. So there are methionine and cysteine, which are sulfur containing amino acids. You can remember it as MSC. S is for sulfur, M which is for methionine, C for cysteine. Okay, then comes another category which includes aromatic R groups. So in aromatic, we have phenylalanine, tyrosine and tryptophan. Okay, so all these three includes benzene ring in their structure. So these are aromatic R groups. Then we have positively charged R groups that include lysine, arginine and histidine. They provide the positive charge in the structure of uh, any protein. Okay, and these positive residues are present in the many important places and performs many important functions. Okay, and some, for example, these are present in the histone protein, positively charged histone proteins, and there are also negatively charged R groups that includes aspartate and glutamate, and these are also present. So these positively and negatively charged R groups are present in the uh, active site of any enzyme. So that is important for the interaction of substrate with the enzyme. So there we can see the role of these positively and negatively charged R groups. So here we have seen all the different kinds of amino acid. So in our question it has been asked, out of these different amino acid, which amino acid is responsible for the intrinsic fluorescence, intrinsic protein fluorescence. Okay, fluorescence. Okay, so if you'll see all the structures carefully, so okay, so before telling the exact answer, I'll tell you that uh, all the different molecules, they absorb a certain frequency or wavelength of light emit and emit at certain wavelength. So that is absorption and emission spectra. So almost all molecules absorb a certain wavelength of the light. Okay, so we all know there are uh, different lights like UV light, X-rays, X-ray light, visible light, infrared light, and so on. So these are the spectra. So here we can say the wavelength of the light increases as we go further, but the energy increases in the opposite direction. Or you can say higher the wavelength, lower the energy of the light. Okay, so all molecules get excited, or in all the molecules the electrons can be excited with a certain wavelength. So uh, in the structure, if you'll see, all can absorb light, but some requires more energy light, that means lower wavelength light, but some require uh, less energy light, that means at higher wavelength they can absorb, or their electron can be excited. So this fluorescence, how it happens, an electron gets excited from ground state to the excited states, and when it returns to the ground state it emits some energy, and that energy can give the fluorescence or the light which can be visualized or can be seen. So in the structure, if you'll see, all amino acids have a kind of localized electron composition in their side chain, except in the aromatic R group containing amino acid that includes phenylalanine, tyrosine and tryptophan. So they all contains a benzene ring in their structure. So this benzene ring is the conjugated electron system. Okay, so here the electrons are delocalized. Okay, so because of the delocalization of the electrons, in these rings, the electrons are being always in moving position. That's why we have the structure of benzene like this, because the pi electrons keep moving, and that's it's called as delocalization of electrons. Okay, so as the electrons are kept moving in these benzene rings or in the aromatic structures, because of their conjugated nature, they are free, or they are more capable to get excited even with the lower energy, because they are never kept free. They are not localized, they are delocalized. So because of the delocalization, they can get excited even with the lower energy and higher wavelength. So these three amino acids which harbors the aromatic benzene ring in their structure, they can get excited at the range of 250 to 280 nanometer. So this is the lambda maxima, or the maximum wavelength at which they get excited. So tryptophan gets excited at the wavelength of 280 nanometer, tyrosine 274, phenylalanine 257. And this is the absorptivity or the molar extension coefficient of these three individual amino acid residues. So here we can see that tryptophan has the highest absorptivity. So the fluorescence or intrinsic fluorescence of any protein comes due to the presence of the tryptophan residue in the peptide chain, because tryptophan gets excited and it absorbs the maximum light and gives the emission or the fluorescence. So majority of the intrinsic fluorescence comes due to the presence of tryptophan in the polypeptide structure. Yes. So for our question, the answer would be tryptophan. We have other options: proline, methionine and histidine. If we look at the structure, so even though proline, but it doesn't have a conjugated uh, electron system, conjugated bonding system here present, so not proline. Another option is histidine. There also not much delocalization of electron as compared to the aromatic R groups or the benzene rings. So not histidine. Other option was maybe methionine. So methionine is also a hydrocarbon chain. So methionine is also a wrong option. So tryptophan, which harbors a aromatic R group in their structure, that is responsible for the intrinsic fluorescence of the protein. And this is my NPTEL reference where you can study about the enzymes in detail. Thank you.